Guys, I want you everybody to be quiet. They're going to start presenting their portion. Students out of your school. Okay, girls, ladies, will you please give the teachers in the front of the class your attention? Ladies, men, Gary, will you please give the teachers in the front of the class your attention, please? You're going to start presenting 13.3.
So 14-3, we're connecting to first vocabulary we're going to have DNA vaccines, which would be a vaccine made from the DNA of pathogen, which would be lethal, but now it's rendered non-lethal.
A telomere is a repeated DNA sequence at the end of the other chromosome that shortens with each cell division.
So what do you know? I know you haven't had a chance to look at the internet, but they're thinking that more research should be done with telomeres. And with telomeres, it might help to slow down the aging process of humans. There's a lot of research and a lot of interest in telomeres right now. Or even the vitamin companies are trying to sell you something that's supposed to make your telomeres last longer. So did you see anything on that, Henry? No? Okay.
For our ethics, this is exactly what it sounds like. It's ethical issues related to biology.
Gene therapy is genetic disorders treated by introducing the gene into patient's cell. So is it the same what the word says, gene therapy?
Okay, so just explain the definitions.
Now we move on to the multiple choice. For any of the pharmaceutical products being produced by DNA technologies are proteins.
When
Can
I
you
give
give me
them,
an example of a protein that they make that's in the book? Yeah, it's, okay. What do they use insulin for?
Helping them mitochondrial sugar.
Well, it helps get sugar into the cell. It helps. It's a protein carrier molecule that helps get sugar into the cell, so the mitochondria can make energy for us. Correct.
when the human body mobilizes its defenses against a pathogen, the body recognizes the pathogen's surface proteins.
Scientists have inserted genes into rice plants that increase the iron and beta-carotene levels.
Okay. So, but Ellen, what if they take that nicely genetically modified rice and they polish it up and turn it to white rice? Do you still get the vitamins from it? The minerals? So, why do you think in a lot of parts of Asia that they always polish the rice and make it white?
They say it's better to eat.
They think it's tastier or something?
And they say it looks better.
So you use the fiber, you use the fat, you use all the vitamins and minerals. So all you're getting is just pure sugar. It's actually a complex carbohydrate, which turns into pure sugar. So it has caloric value. So you get calories from it, but you don't get the nutritional value other than calories. Allison?
Some people are concerned that genetically engineering companies would transmit their new genes to development species, producing super roots.
So what would happen if we had super wheat? How could that create a problem? Class. You know, Allison doesn't have to necessarily answer. Does anyone want to answer that? What happened if they genetically modified a plant and then the wheat was next door and the wheat somehow got that modification and it produced a super wheat? How could that create a problem? Okay, that's good. Eddie? So why, you guys, has anybody ever had a garden or worked in a garden? Does anybody has a garden at their house or worked in a garden?
Yes.
I have a garden. I never
So-
had it.
So do you know that, like, you plant these really beautiful plants, you water them, you fertilize and take care of them, and the weeds overgrow and kill them. And you're always pulling out the weeds, and you're never pulling out your plants very often. Why do weeds work so much better than the fancy plants we put in and we try to plant and grow? Why do you think weeds work better? We're going to be going into evolution in one of our next chapters coming up. So why are weeds tougher and live easier than all these fancy plants we grow?
They absorb fertilizer.
They have evolved so they can live on less water, less sunlight, and don't need as much nitrogen and a lot of different factors. So weeds have evolved to be tough, and they don't need anybody to take care of them, they can take care of themselves. A lot of the plants we want to grow are special, beautiful plants, or they're fruit-producing plants, and those take a lot more energy. And we have to work a lot harder to keep them going than you do a weed. I mean, I was doing a video for you guys in audio on photosynthesis, and when I was recording, I noticed that there was a weed hiding behind a big, pretty tree I planted. It was getting no sunlight at all, except for reflected sunlight and no water, and it was growing perfectly. So that's kind of interesting, but it has to do with evolution. Okay, keep going. Sorry for that.
To move on to short answers, how does a DNA vaccine prevent future disease? The DNA directs the synthesis of proteins. The vaccine is injected into the patient, and the body creates antibodies to protect against proteins.
Okay, so if you inject somebody with an attenuated virus or an attenuated protein that they've made so it's weaker, it'll still have the genetic code of it. So our body will still make antibodies to it, but you won't actually get the sickness. So it could be like a virus that could cause a flu. But it's a weakened virus. It's been heat treated. Somehow they attenuated it so it's no longer pathogenic and no longer make you sick. Cause pathology. But it still has the same genetic code, so we can still make antibodies to it. So if we get exposed to that virus that's strong and healthy, we already have the code to make the antibodies so we can fight it off and have more resistance. You guys understand that part? How that works? Okay, Howard? Or Henry, who's over next? I'm sorry.
If Dolly's clothing was successful, why was her lifespan shorter than usual? She suffered from premature aging. Nuclei chromosomes shortened 12 years.
So what happened with the telomeres?
It got shorter.
Shortened the telomeres, so she aged premature. Okay, Helen?
Why doesn't gene therapy cure cystic fibrosis? Gene therapy, the virus only goes on the surface, but actually the virus carrying the normal gene should reach the deep amount of cells to cure cystic fibrosis, where the common gene is most needed.
Okay, okay. So, and it also talks about how it goes into the nucleus of the cell, and they can't get to do that, so they can't kill it. So they can help cystic fibrosis, but they have to keep getting the gene therapy because it doesn't stick because of that. Allison?
And describe the potential problem that could arise from genetic aging. The site resistance can be positively insulin.
So I'm sorry, I couldn't quite hear you. I didn't understand what you said. Can you say that again a little louder, please?
So potential problem that could arise from genetic
Engineering.
is weeds can become super.
Wheat, like wheat bread?
We could use
Oh, weeds can become super weeds. I'm sorry. I couldn't hear what you were saying. Okay.
Now we're on to structures and functions. So, from genetic engineering, we move on to animals mass produced. You have A for medicines, and B, organs that don't trigger rejection. Then we move on from genetic engineering down to plants that produce a pest-resistant crop for A, and a herbicide-resistant crop for B.
Okay, good. guys all have copies of this? You guys all have copies? Okay. You girls done? You girls ready to present? Who's gonna get it? There was 3 things, there was 13, did you do 13 1, 13 2, or 13 3? These guys are 13, which group are you guys in, what number? Okay, go ahead and get started.
So we're gonna present about the Human Genome Project. First vocab that we have is proteomics, the study of all of an organism protein, including their identity, structure, interaction, and abundance.
Abundance, what are you saying, I'm sorry? Abundances, how much there are, okay.
And the second one is bioinformatics, the use of computer to investigate biological questions. Another word is single nucleotide polymorphism, the unique spots where individuals differ by a single nucleotide. There are 8 million NNPS in the
Genome.
Human Genome Project is a research effort to sequence the entire human genome and locate all the functionally important sequences.
Now, is that still, is the research still be going on in the Human Genome Project? Okay, good.
So one of the goals of the Human Genome Project is to determine the nucleotide sequence of the entire human genome. One of the surprising discoveries of the Human Genome Project was that the human genome consists only about 30,000 to 40,000 genes. And an understanding of the human genome is aided by the understanding of the genomes of model species. Only about 2% of the human genome codes for proteins. DNA microarray is an important tool because it shows which genes are active in a cell.
So what the scientists want to map the human genome, they want to know the location of all the important genes and how genes expression and cell growth are controlled and about human evolution.
Human what?
Evolution.
Evolution, okay, good.
The three important discoveries resulting from the Human Genome Project are they found out that only 2% of the human genome encodes protein, and then exons are spliced to allow a gene to encode different protein, and human genome contains about 30,000 to 40,000 genes. And so why is it more important to understand the human proteome than the human genome? Because it is the protein that carries out the work of the cells, and then scientists must understand how protein work in order to understand how genes work.
What is the relationship, if any, between the complexity of an organism and the size of its genome? Like the more complex an organism, that is, the larger genomes and less complex organisms. Like, second question is like, what might explain why there is not an indirect relationship between the size of an organism and the genome and the number of genes it contains? Like, which there's no such thing as a relationship between these factors. Like one of the examples is that a mouse, which has like 30,000 genes, and a human has like 35,000. Like they almost have the same amount of genes, and like even though a human is more complex than a mouse in their structure, the function, and the size.
Now what about like the cerebral cortex on a human versus a mouse?
Sorry, what?
The cerebral cortex, the part of the brain that we think with, is ours a little different than a mouse's?
Yes.
Yeah, but it's interesting that we don't have that many more genes than they do. So there is no direct correlation between the genome and how complex the animal is. Okay? Are you guys done? Good job. Okay, the last group. Did you ever bring attention to the first 20 minutes of class, like weight group, everything? Already? You guys are paying attention to everybody? You guys will pick up
So DNA fingerprint. What is DNA fingerprint? It's a resulting pattern of mechanisms. Oh, good stuff.
Keep going, guys.
So DNA fingerprint is a resulting pattern of gel electrophoresis is like a technique to separate nucleic acid and protein accordingly based on their size and charge.
A probe is a strand of RNA or a single-stranded DNA that's like labeled with a radioactive element or a fluorescent dye, and it can be basic paired to a specific DNA. Primers artificially make pieces of single-stranded DNA that is 20 or 30 nucleotides must be present for DNA polymerase to initiate replication.
So moving on to the multiple choice. To cut DNA molecules into pieces that have specific sequences of nucleotides, genetic engineers use precision enzymes in order to cut DNA molecules. In gel electrophoresis, DNA fragments migrate toward one end of a gel because they are attracted to the positively charged end of the gel.
The accuracy of DNA fingerprinting can be increased by comparing reprints that only one or two sides. The accuracy of DNA fingerprinting can be increased by comparing non-coding segments from
Several different loci. And the incidence, if they do like 7 loci or 10 loci, it's 1,000,000 to 1 chance that those two people, if they match, that they would not get the same. So the more loci they do, the more accurate the DNA fingerprinting is. If they go over 7 to 10 loci, it's actually 1,000,000 to 1 odds that that's not the right person or not. So it makes it much more accurate the more loci they do. Go ahead.
In addition to DNA polymerase and primers, the polymerase chain reaction also requires a supply of the 4 DNA nucleotides.
To obtain bacteria that produce insulin, genetic engineers insert a factor containing the human gene for insulin into the bacteria.
Okay, so what did they first put it into? What's that thing called, that brown thing? That they first, yes, plasmid, okay. And then what do they call it when it has bacteria and human together? What's that called? Recombinant DNA.
There's a short answer. The question is, how are radioactive probes useful in DNA fingerprinting? So when viewed under the ultraviolet light or exposed to a photographic film, the clone of the cells bury the donor DNA and is attached to probe lobes and reveals its location.
How is the polymerase chain reaction useful in DNA fingerprinting? PCR is the technique that quickly produces many copies of DNA fragments. So if that is done, DNA fingerprinting can be done so that they can get enough DNA fragments.
Yeah, so they may not have enough cells, like I said, is the guy was smoking a cigarette and only got a few skin cells from his lips. There's not enough to do it, so they'll do PCR, and when they do it 30 times, 30 divisions, there'll be over 1,000,000 new DNA fragments made. So we'll have plenty for the gel electrophoresis.
Why is it necessary to use the same restriction enzyme to cut two pieces of DNA that are to be joined together? So the
And what do they call that when they cut in the same way? Sticky ends. Okay, go ahead.
So these three ways that DNA technology could be used to improve the life of humans. Firstly, DNA technology can be used to cure diseases, to treat genetic disorder, to improve food crops, and many other things.
Excellent. So, Reed, what problems do we have with genetic crops sometimes? It's genetically modified, like genetically modified corn, soy, or wheat. What problems could that cause? Even though they're trying to make the crops healthier and better and more nutritious for us, what problems have they noticed? And it's not in your book, but I've talked about it lots of times. What problems are we having with genetically modified soy, corn, and wheat?
Well, like a lot of companies modify them too much, so then our body doesn't recognize that.
Recognize the what in the wheat or the corn? The
Protein.
protein, yeah, because we're talking about genetic engineering of proteins. And what happens when our body doesn't think it's a normal protein, it thinks it's an invader? What does our body do with that? And what problems does that cause to start with? Us not accepting protein. What would our body do if they had a foreign invader? How would it defend against a foreign protein? Does anybody know that question? If our body had a foreign protein in it, like a virus or a bacteria, that it thought was a pathogen, what would our body do to respond to that? The immune system would be triggered and would make white blood cells to destroy it. Now the problem with genetically modified corn and soy and wheat is our body reacts to that, it ramps up our autoimmune system, and wheat in particular, we've been eating it for 4,000 years, and the first 25 amino acid sequences is the same as human brain tissue, human thyroid tissue, and human GI tissue. So it attacks the GI tract and causes irritable bowel syndrome, Crohn's disease, malabsorption syndrome, it destroys the microbiota. Instead of being like this, they go like this, and you don't get absorbed nutrients. It attacks the brain, causing Alzheimer's, dementia, and Parkinson's. It also attacks the thyroid. The most common cause of Hashimoto's or autoimmune thyroid disease is eating too much wheat, especially wheat combined with dairy, because they're cross-contaminants, and they make each other more antigenic and more reactive in our body. Okay, good job, guys. Thank you.